I'll start us off with an overview of 10x Genomics single-cell gene expression 3' v3.1 dual index solution. So first off, let's discuss why we perform single-cell gene expression. What advantages does it offer over bulk analysis? Single-cell RNA-Seq allows researchers to resolve the complexity of cell types and states present in diverse samples to a degree not possible by traditional bulk RNA-Seq methods. This is because during bulk RNA-Seq, all cells in a sample are lysed and gene expression is captured at the sample level, while single-cell RNA-Seq allows the transcriptome of individual cells within a heterogeneous sample to be profiled. The Chromium single-cell gene expression solution with optional feature barcoding technology enables unbiased gene expression analysis at the single-cell level. Feature barcoding technology provides simultaneous assessment of perturbations in the case of CRISPR and quantitative analysis of protein abundance in the case of cell surface protein applications. This assay can also be performed with cell multiplexing or targeted gene expression. The solution is ready to use, has a robust workflow, and is compatible with demonstrated protocols for multiple sample types, including cell lines, primary cells, and dissociated tissue. It's compatible with whole cells and nuclei. The solution also includes turnkey analysis using our Cell Ranger analysis pipeline and visualization software through Loupe browser. The solution is enabled by our proprietary gel beads. Each gel bead contains many copies of 3 types of oligos, each having the same barcode, but a unique UMI for every individual oligo. Each oligo consists of a partial TrueSeq read1 sequence for Illumina Sequencing, a 16 nucleotide 10x barcode, which identifies the particular cell in which a transcript or feature is found, a 12 nucleotide unique molecular identifier, or UMI, which is used to identify the particular molecule that is present in your library, and a poly-DT sequence that enables the capture of polyadenylated mRNA molecules. Thus, any polyadenylated mRNA molecule can be captured using this solution. To enable the feature barcoding technology, 2 additional oligos are present on the gel beads, which contain capture sequences for features of interest. The features may be oligonucleotide conjugated lipids for cell multiplexing, oligonucleotide conjugated antibodies for cell surface protein profiling, or single guide RNAs for CRISPR experiments. Single-cell capture is performed using our single-use microfluidic chips that allow partitioning of individual cells with 10x barcoded gel beads into droplets. Up to 8 channels can be run in parallel. Each channel can accommodate between 500 to 10,000 cells per sample using the standard assay. The partitioning on the Chromium X series instrument, or Chromium controller, only takes 18 minutes. During partitioning, up to 65% of input cells are captured. This means that loading approximately 16,000 cells for one sample will yield data from approximately 10,000 cells. While most occupied gems will contain a single cell, there is a statistical probability that some gems may contain more than one cell. The expected fraction of droplets with more than one cell increases linearly with the number of targeted cells. For 1,000 cells, the multiplet rate is 0.8%. During the workflow, single-cell suspensions are combined with master mix and loaded onto the Chromium chip, along with 10x barcoded gel beads and partitioning oil. The chip is placed in the Chromium X series instrument, or Chromium controller, where the gems, or gel beads and emulsions, are generated. Most gems will contain a 10x gel bead, while a subset will also contain a single cell. After the gems are generated, the gel beads dissolve and the cells lyse, releasing the oligos and mRNA into solution. Within the gems, reverse transcription occurs, which converts the mRNA and/or the features of interest into 10x barcoded cDNA and feature barcode DNA. After barcoding, The gems are broken open, and the barcoded fragments from a single gem well are pooled together. The final library construction steps are performed in bulk to generate a final library ready for sequencing. Final library constructs are sequenced, and the 10x barcodes are used to associate individual reads back to individual partitions and to each individual cell. The resulting output is a digital single-cell gene expression profile with information on your feature of interest. So now that we've reviewed how the assay works and the overall workflow, I'd like to discuss best practices for generating single-cell gene expression libraries. The focus of this webinar is on library preparation, so I will only highlight a few key considerations for sample prep. We have a variety of demonstrated protocols for preparing different sample types on our support site, as well as additional technical notes and webinars devoted to sample prep that provide more in-depth recommendations. Good libraries start with high-quality samples. In order to generate single-cell data, the cell membrane must remain intact to prevent nucleic acids from leaking outside the cell. Ambient RNA in the suspension will get captured in gems and can contribute to background signal that can complicate analysis. Before using samples for single-cell gene expression, we recommend performing a few quality control checks to confirm that the sample has not been compromised. We recommend checking the viability of the sample using AOPI or trypan blue staining to confirm that the cells are viable. Ideal samples will have over 90% viability, but the solution is compatible with samples with over 70% viability. If viability is lower than 70%, use of a dead cell removal kit, FACS, or gentle centrifugation is recommended to remove dead cells and debris. Having an accurate cell count is important to ensure that the desired number of cells are being analyzed. Loading significantly more cells than expected can increase the multiple rate or increase the chance of a microfluidic clog. Either a cell counter or hemocytometer can be used for cell counting. Taking multiple measurements is recommended to ensure that the count is representative of the sample. The final cell suspension should be diluted in a compatible buffer to a final concentration that is within the optimal cell stock concentration listed in the user guide in the cell suspension volume calculator table. Examining the single cell suspension under a microscope is important to check for debris or cell clumps that can clog the microfluidics. If debris or clumping are identified, running the sample through a filter is recommended. Lastly, please load the sample on the chip within 30 minutes of preparation, as cells in suspension can lose viability quickly. Once a single cell suspension has been generated, the first step is to combine the suspension with the master mix and load onto the chip. Careful attention to the steps in the user guide is essential to avoiding failures in generating gems. I'd like to highlight a few of those steps. The first is equilibrating the gel beads to room temperature for at least 30 minutes before loading on the chip. Be sure to resuspend the template switch oligo in low TE. EDTA can inhibit the reverse transcriptase reaction and result in low to no cDNA being detected if regular TE is used. Always add water to the master mix first, and then add cells to the master mix to avoid lysing the cells. Load the chip in the order listed in the user guide, and immediately remove the gems once the run is complete. Only use supported plasticware and tips, as some plastics can increase the likelihood of wetting failures. While transferring the gems from the chip to the PCR strip tube on ice, visually inspect the gems to confirm that the emulsion is opaque and uniform. Take a photo at this step for future reference in case of unexpected results or for use if submitting a warranty replacement claim if a gem generation failure is detected. In the image on the right, pipette tips B, D, and F have a uniform and opaque appearance, and illustrate what successful gem generation looks like. Pipette tip C and E are examples of wetting failures, indicating a loss of partitioning of single cells. The emulsions are more clear and non-uniform. If a wetting failure is observed, contact 10x Genomics support and re-prepare the sample. Pipette tip G has a uniform emulsion, but less than the full volume of emulsion is recovered. This indicates a clog. It is recommended to repeat any samples that have a clog. However, if the sample is precious and cannot be repeated, it may be possible to generate single-cell data by continuing. Please contact 10x Genomics support if either a wetting failure or clog are identified for additional guidance and to request replacement reagents. As part of our warranty policy, clog and wetting failures that have been properly documented are reimbursed with replacement reagents and chips. At this point in the workflow, the single-cell suspension has been loaded onto the chip with the gel beads and reagents to generate gems. Inside each gem, the cells are lysed, and the polyadenylated mRNA transcripts are captured by the poly-DT primer on the gel bead oligos. Once the gems have been collected, they are incubated on a thermocycler for reverse transcription. The template switch oligo in the master mix allows template switching to occur, followed by extension of the transcript so that each cDNA is now labeled with a partial read1 sequence, a 10x barcode to identify which cell the transcript is associated with, a unique UMI sequence, the insert sequence, and the TSO sequence, which will be used for cDNA amplification. Once the RT reaction is complete, the gems are broken open and all the transcripts from a single gem well are pooled together for the remaining library steps. Once the cDNA is generated, it is amplified using primers to the read1 and the TSO sequences. The targeted cell recovery is used to determine the number of cycles for the cDNA amplification. Inaccurate cell counts can lead to over or under amplification, which can result in PCR artifacts or insufficient yield. After the cDNA amplification, there is a single-sided size selection and cleanup using SPRY Select. Be sure to follow our best practices for SPRY Select cleanups, which include preparing fresh 80% ethanol each day, ensuring that the SPRY Select is fully thawed and equilibrated to room temperature, vortexing the SPRY Select reagent thoroughly before adding to samples and ensuring the samples are fully mixed with the beads, avoiding transferring excess SPRY beads that may have stuck to the outside of the pipette tip, making sure to remove all of the ethanol before resuspending the bead pellet, while also not over-drying the bead pellet. Failure to follow these best practices can result in an incorrect size selection. Once the cDNA is amplified and cleaned up, it is important to confirm that the fragment distribution and yield are as expected before proceeding. This can be done on a bioanalyzer, tape station, lab chip, or fragment analyzer. Be sure to load the sample within the concentration range for the method used. In most cases, this can be achieved by using a 1:10 dilution of the cDNA. Over or underloaded traces may result in inaccurate yield estimations. Since the cDNA yield is used to determine the number of cycles for the sample index PCR, an inaccurate yield estimation can lead to PCR artifacts for the sample index PCR. Fragment distributions and yields will vary depending on sample type. Check for signs of mRNA degradation. Most mRNA fragments are expected to range from about 500 base pairs to a few thousand base pairs. Short degraded fragments can lead to a lower transcriptome mapping rate. If significant smaller fragments are detected or the yield is unexpectedly low, contact 10x Genomics support for assistance with optimizing sample prep. One thing to note is that final libraries can be generated that have the expected fragment distribution from samples with poor cDNA. However, the resulting data will likely be compromised. So checking the cDNA quality is highly recommended, as the additional size selection steps during library construction can mask issues with the underlying cDNA if only relying on the final library trace for sample quality control. After confirming the cDNA quality, you can proceed with library generation. 25% of the cDNA will be used for library construction. The remaining cDNA can be saved for generating additional libraries. Enzymatic fragmentation will fragment the cDNA to generate fragments that are the optimal length for Illumina sequencing after additional spray size selection. Be sure to pre-chill the thermal cycler block to 4 Celsius prior to assembling the fragmentation reaction to prevent over-fragmentation. End repair and A-tailing will prepare the fragments for the read-to adapter ligation. Prior to the ligation, a double-sided spray cleanup is performed to remove fragments that are smaller or larger than the desired length for sequencing. Afterwards, the read-to sequence is ligated onto the end of the insert, and a single-sided spray cleanup is performed. The sample Index PCR adds the P5 and P7 adapters required for clustering on an Illumina flow cell, as well as the I5 and I7 index sequences used for demultiplexing. Dual Index Kit TT Set A is used for this solution. Record the well ID used for each sample, as this is needed for demultiplexing. Incubate in a thermal cycler using the appropriate number of cycles, depending on the amount of cDNA used for library preparation. A final double-sided spray size selection is performed to clean up the sample. Confirm that the final library is between 300 to 700 base pairs by running on a bioanalyzer or tape station. The average fragment size should be about 450 base pairs. If smaller products are observed, an additional spray cleanup can be performed. Contact 10x Genomics support for recommendations for the spray ratio to use. Library quantification should be performed using the Kappa Library Quantification Kit for Illumina platforms, as outlined in the appendix of the user guide. The Kappa qPCR will only quantify fragments containing both a P5 and P7 adapter, which provides a more accurate quantification of sequenceable fragments compared to methods that measure all double-strand DNA. Dual-index single-cell gene expression libraries can be pooled with other 10x dual-index libraries, including the standard LT or HT 3'V3.1 assays, the standard or HT 5'V2 assay, Visium for fresh frozen samples gene expression libraries, and multi-omi gene expression libraries. We recommend sequencing any feature barcode libraries with the gene expression libraries. Alterations to the cycle numbers may be required when combining different library types, so please confirm the sequencing specifications listed in the user guide or on the support site. Ensure that index sequences from pooled libraries do not overlap. The libraries can be sequenced on a variety of Illumina sequencers listed at the bottom right. The recommended loading concentrations can be found in the sequencing section of the user guide. Read 1 consists of the 10x barcode and UMI and requires 28 cycles. The I5 and I7 index sequences should both be sequenced to 10 cycles. Read 2 consists of the cDNA insert and should be sequenced for 90 cycles. We recommend a minimum of 20,000 read pairs per cell as a starting point for sequencing. Depending on the complexity of the sample and the research goals, additional sequencing may be required. And I'll now turn it over to Judy to discuss considerations for setting up the sequencing run.
Thank you so much, Christine, and thanks everyone for being here today. Here's a list of the supported sequencers, which are the sequencers that we have officially tested the libraries on, but chemically speaking, the single-cell libraries are compatible with all Illumina sequencing platforms. The different instruments require the I5 index to be in different orientations depending on the software used. So this is something to keep an eye on when you're setting up your run. Adaptive trimming is not a setting that we want to have during FASTQ generation. The slide says it's not required, but it actually is something that we really don't want to have because it could interfere with the downstream analysis. And because the I5 and I7 indexes are barcodes that are used for demultiplexing, standard FASTQ generation with any Illumina software can be used. One additional note for those of you who may be familiar with with short read masking, this is something a process where reads of a certain length get converted to a string of ends if they are especially short. But our shortest read length for read 1, which is 28, is longer than the default value that triggers this from happening. So we do not need to adjust any settings in that regard. How many samples can you load for sequencing? Here is a table that provides you with a good starting point for some of our sequencers. Here the NextSeq 550, the NextSeq 2000, and the NovaSeq 6000. So this is a good point to start, but again, as usual, this might require a little bit of optimization depending on your setup and your quantification method. And on the bottom is again a reminder of the read length to set up your run that Christine had already shown in one of our previous slides. So let's talk about the demultiplexing workflow. So demultiplexing is the process that converts our raw sequencing data as it comes off the instrument in the form of BCL files to FASTQ files. These are then separated per sample and per week and are used for the further downstream data analysis to give you your interpretable results. The demultiplexing can be done in several different ways, so we want to spend the next couple of slides talking about that in a little bit more detail. There are a lot of options, and there really isn't one option that is the best one or that we would recommend over the others. It just depends on your setup, your lab, and what works best for you. So in general, demultiplexing can be done on a server or on the sequencer itself or the sequencers that support this. It can be done on BaseSpace Sequence Hub, or it can be done on the 10x Genomics software Cell Ranger. And then the FASTQ files are further analyzed in Cell Ranger to give you your results. So let's talk about the first scenario where we would set up our run in manual or standalone mode. This is an option for all of our sequencers, and it generally does not require a sample sheet for the run setup. The only exception is the MySeq with MySeq Control Software 2.6. That does require a sample sheet for setup, but any other higher control softwares and all other instruments do not require this. So you would then get BCL files from your run, and those can be analyzed in two ways. So you could either use them as direct input into Cell Ranger, or you can use them on a server running BCL to FASTQ or BCL convert to generate your demultiplexed FASTQs and then input those into Cell Ranger. So Cell Ranger can accept either the BCL files as input or the FASTQ files. So on the sequencer itself, all you have to do would be to set the cycle length and the output location and start your run, although attaching a sample sheet is optional. And at the bottom of the slide, there's a link to the 10x support site for some example sample sheets and BCL to FASTQ commands. Our scenario number 2 is using BaseSpace Sequence Hub upload. This is also an option for all of our sequencers, and it does require a sample sheet in some shape or form, so either attached manually or auto-generated by LLM, IRS, or PrepTab. And it generates the demultiplexed FASTQs that can then be downloaded from BaseSpace and used as input into Cell Ranger. So this is a good option. It's very convenient for storage, for example, to store your FASTQs in the cloud, or you can use them for other applications on BaseSpace. So if there's any other programs that you might want to use to analyze your data that's conveniently already located in BaseSpace. Your option number 3, or scenario number 3, is the on-instrument FASTQ generation. Now, this is only possible for the supported sequences, which include the NextSeq 1000, 2000, the NextSeq 550, if you are on control software 4, the MySeq, and the iSeq 100. That gives you the demultiplexed FASTQs as an output, also requires a sample sheet, and then those FASTQs are used as input into ScellRanger. Now let's talk about the sequencing run evaluation. So now that we know how to set up our run and demultiplex our data, how do we know if a run is actually successful once it's complete? So we can evaluate sequencing runs in the program Sequencing Analysis Viewer or on BaseSpace Sequence Hub. Illumina has instrument specifications for different sequencers and read lengths, and those can be found at the link provided on this slide. In general, important metrics to assess are the total data output in gigabases, the percent of bases caused with a quality score of 30 or higher, or Q30 score, the clusters passing filter, and the percent aligned, which refers to the amount of phi-X that was added with your run if you decided to use phi-X. So now let's dive into 2 example runs of single-cell gene expression libraries. The first one here is sequenced on the NextSeq 2000. These are actually example runs that can be downloaded from BaseSpace Sequence Hub from our demo data, and you can find details at the bottom of the slide. So let's start by actually looking at the different fields that we see here in SAV in general terms. So here on the left, we have the flow cell chart, which shows the color-coded metrics per tile for the entire flow cell. Here, the data by lane, which is something that we often look at. It displays the various metrics for each cycle of the run, and they can be selected using the drop-down list. Here is the Q-score distribution, which shows a quick overview of the quality of the run, and you can find the Q30 for the whole run in the upper right of this box. The data by lane shows the plots of metrics per lane, and then here we have the Q-score heat map, which shows a heat map for the Q-score by cycle. If we switch over to the summary tab, here we can see the total yield for the run, which is 75 gigabases for this run. Here we have the Q30, which is 95%. So that's a really good value for these libraries on the NextSeq 2000. If this value for any reason is lower than expected, you can look at the individual Q30 scores for each read, and this can help us determine if there's a particular read that might be causing the overall Q30 to be calculated lower than expected. But in this case, they're all nice and consistent for each read. Here we have the clusters passing filter value, another really good quality control value, and it's in the mid-80s for this library. And then here the percent aligned value refers to phi-X that was mixed in with your library for the run. Now what we're looking for here is generally to see a percentage that roughly corresponds to the amount that you have spiked in with your library. 1% is recommended, but this can be adjusted as needed. And if the value is drastically different compared to what you had added with your library, that can indicate possible issues with mixing, denaturing, or loading. Going back to the analysis tab, if we look at the data by cycle and flip the drop-down to Q30, we get an even more granular view of the Q30 value for each read really across our 4 reads, each cycle of the 4 reads. And so this can help us analyze again if our overall value is lower than expected. Is this where is there a sudden drop somewhere in which read? So this is a good tool for analysis. If we switch the data by cycle to percent base, this gives us the characteristic profile for this library. And so what this shows us is the percent of AC, G, and T base at each cycle of each read. And the shape of this plot really corresponds directly to the structure of your library. So this is a good snapshot to see that your just overall percentages of bases look as expected. Another really important thing that we use to diagnose potential causes for lower than expected quality metrics is over or underloading. So this is a little different depending on the sequencer that you're on. So the way we do this on the NextSeq 2000 is by assessing the percent loading concentration metric. This can either be seen in BaseSpace under the metrics tab, which is shown here on the screenshot, or you can check in your completed run folder and look for a file that is called primaryAnalysisMetrics.csv, and that will contain this value. For most libraries, we want to see a value between 95 to 99%. And if the loading concentration is too low, you can increase it in 100 picomolar increments over subsequent runs. Okay, let's look at our second example, which is single-cell libraries on the NovaSeq 6000. Here we are on the Analysis tab, looking at the data by cycle. Flipped to the Q30, we can again see that our Q30 scores or values stay nice and consistently high across all the 4 reads, and there's no real unexpected drops here anywhere. If we go to our summary tab, we see that our overall yield is 132 gigabases, and our Q30 score is 91. So that's a really good value. And looking at our 4 reads, it also stays nice and consistent. Looking at the clusters passing filter, it's in the mid to high 70s. So this is also really excellent for these libraries on this sequencer. And here looking at our percent aligned with this one, we spiked in 1% 5x. So we see it stays really close to that value, and no indication of any loading issues. How do we determine over versus underloading on the NovaSeq? So that's a little bit different. Here we want to plot the percent occupied by the percent pass filter values. Now we have a really good step-by-step guide on how to do this, and there's a link at the bottom of the slide. But basically, you would open the Imaging tab in SAE, or in Sequence Analysis Viewer, and create a scatter plot of percent pass filter and percent occupied, and then evaluate its shape. An underloaded library generally shows a diagonal line. An overloaded library presents as a vertical line, and optimal loading is shown when you have an oval shape, such as presented here on this slide. Here is another summary table that contains some suggested loading concentrations and typical sequencing metrics for several sequencers for the Chromium single-cell library. And so this is a great place for you to start in regards to loading. Might require a little bit of adjustment depending on your quantification in your lab, but it's a good starting point. And then we have some specifications for percent Q30 and yield per lane that you can use to compare your run values to. Okay, and now for considerations for analysis, I'm going to pass it back over to Christine.
Thank you, Judy. So ScellRanger produces a web summary for every run that has many metrics that can be used to assess the quality of the run. And I'd like to highlight a few of these metrics briefly that can be reflective of the sequencing quality. The valid barcodes refers to the fraction of reads with barcodes that matches our whitelist of known barcodes after any barcode correction. The fraction of reads with valid UMIs lists the fraction of UMI sequences that do not contain any ends or no-call bases and are not homopolymers. If either of these metrics are low, there could be an issue with either the reverse transcription step or with the sequencing run. The Q30 bases in barcode, RNA read, and UMI list the fraction of bases that have a quality score greater than or equal to 30. Lower quality scores can result in a portion of the reads being unusable. This may indicate issues with the library preparation or with the Illumina sequencing quality. Reads mapped confidently to transcriptome indicates the fraction of reads that map to a unique gene in the transcriptome. The read must be consistent with annotated splice junctions. If this metric is low, it may indicate use of the wrong reference transcriptome, a poorly assembled reference, poor library quality, poor sequencing quality, or reads that are shorter than the recommended minimum. And the fraction of reads in cells refers to the fraction of reads that contain a valid barcode, are confidently mapped to the transcriptome, and are associated with a barcode called as a cell. If this metric is low, there are high levels of ambient RNA partitioned into all gems, both cell-associated and non-cell-associated gems. This provides an indication of the level of background signal in the sample, and sample prep optimization is recommended. For additional information about interpreting web summaries, you can view our Interpreting Cell Ranger web summary files for the single-cell gene expression assay tech note that we have on the 10x Genomics support site. So now that we've covered best practices for library preparation and how to evaluate the sequencing run, Judy will discuss the support partnership between 10x Genomics and Illumina that we've developed to make resolving any issues that you could encounter faster and easier.
Yeah, so before ending the webinar, we just wanted to tell you about our support collaboration. The first thing that I wanted to note is that we would never share any data or even any customer information that is shared with either Illumina or 10x Genomics without specifically asking for your consent first. But what we're trying to do is really just make the process a lot easier for you. If you've ever worked with or used a product that had an issue that was supported by two different companies, you may have found yourself in a situation where you call company A and explain the whole issue, and they do some troubleshooting, and then they say, oh, it's not with us, call company B. And then you have to call them up and explain everything all over again, and then they might send you back to company A and so forth. And we really want to really make this process easier for you. We want to make sure that information does not have to be repeated over and over. We want to do more efficient troubleshooting by talking amongst each other to determine if the root cause may be with an Illumina product or with a 10x Genomics product, and then give you just a faster resolution.